Greetings, ladies and gentlemen, and welcome to the Chemistry of Organic Molecules. I have my usual meme or joke on the first slide. Read at your leisure. If you get it, you are an awesome nerd. If you don't get it, don't worry. There's still time to become an awesome nerd. So let's dig right into this. Organic molecules, also sometimes called biomolecules, also sometimes called macromolecules, are the molecules that make up the vast majority of our body. Not so much by mass, because that would be water that makes up a large amount of our body, but they do make up a lot of the parts of our body that give us the unique properties of living things, as well as help our cells to function. So there are four major categories of these guys. They all contain carbon and hydrogen. There are carbohydrates, lipids, proteins, and nucleic acids, and we'll talk about each of them throughout this lesson. First, let's clarify what an organic molecule is. They will always contain carbon and hydrogen, and they are always covalently bound together, so they're going to be sharing their electrons. Some of them can be very large with thousands of atoms, but some of them can be smaller. For example, the simplest organic molecule is this guy right here. Very tiny, only 5 atoms, that's methane. Or you can have these organic molecules that are so large that, they span an entire cell. Organic molecules are usually associated with living things, but we're going to talk later about one of the kind of strange aspects of the nature of organic molecules, and that is they had to come from inorganic molecules at some point. Inorganic molecules are usually going to be smaller in size. There's often ionic bonding that's taking place, and that means that they would contain positive and negative ions. So, in addition to carbons and hydrogens, there are a lot of what are called functional groups that can be attached to the backbone or the carbon skeleton that makes up the center of these organic molecules. So there are a number of these functional groups. Each of them can provide different characteristics to the molecule that they are a part of. For example, a lot of these confer polarity. So the hydroxyl group, or OH, is polar, and it forms hydrogen bonds. The carbonyl group is also found in some sugars, as is the ketone group. Notice that when there's an oxygen present, oftentimes that is going to confer the polarity to a molecule. Carboxylic acid is a part of an amino acid, and then the other part of the amino acid is an amine group, which is also polar. Phosphates are very important because not only are they polar, but they are super important when we talk about energy and also when we talk about DNA and RNA. And then when we talk about protein structure, an important but not always mentioned functional group is the sulfhydryl group. It's a sulfur and a hydrogen. And this is important because in certain proteins, these sulfhydryl groups will actually, you'll have like a sulfhydryl group sticking off of one amino acid, you'll have a sulfhydryl group sticking off another amino acid, the hydrogens will come off, and the two sulfurs will bond to each other and form this strong covalent link between two different parts of a protein that really helps stabilize the structure. So, when we're talking about these complex molecules, they are often, most of the time, made of repeating subunits. These repeating subunits generally have similar structures, similar shapes, and they're called monomers. When you put many monomers together to form a larger molecule, that creates a polymer. The way I tend to think of it is a monomer is like a single Lego brick. A polymer is when you take those individual Lego bricks, you put them together to create some completely different form. Depending on the nature of the monomers, you might be able to make thousands or hundreds of thousands of different forms, like what you get with protein. Or if you have a monomer like a nucleotide, well, there are some limitations to the structure of nucleic acids. So Your example, amino acids are the monomers of proteins, which are a polymer. So obviously polymers are going to be bigger than their monomers. There are a couple of other terms. Dimer is two monomers bound together, and you'll run across that term if you're looking at some biochemistry information. You can also run across the term oligomer. And an oligomer is a couple of monomers bound together, more than two obviously, but not anywhere near 10 or 20 or 30 monomers bound together. So a lot of monomers are going to have an OH or some sort of oxygen group, maybe a carbonyl or a carboxyl group, and then they're going to have a hydrogen on the other end. And this is important for when we talk about how these molecules bond to each other. So there are two important sets of reactions we're going to look at: dehydration reactions, sometimes known as dehydration synthesis, other times known as condensation reactions. Just note those are all three synonyms for each other, are the processes of building polymers. So this builds, and the other process is the opposing process. It's called hydrolysis. If you break the term down, what does hydro mean? Well, hydro means water. And if you remember what lysis means to break or burst. So it literally means breaking apart using water. So the components of a water molecule are separated from each other and used to break apart the covalent bond that is holding two monomers together. So while dehydration reactions and dehydration synthesis builds polymers, hydrolysis breaks polymers down. So we're going to look at an example of this happening. We have two generic monomers. Notice that each of them has an OH on one side and a hydrogen on the other side. So what can happen is there are enzymes that will help break off a hydrogen group from one and an OH group from another. When that happens, that hydrogen and that OH can combine to form a water molecule. That is the dehydration aspect, because you're taking away the equivalent of an entire water molecule from these two monomers. Then the synthesis part is joining these two monomers together. So you get a dimer and you get a leftover water molecule. And this is, one reason why water is really essential. It forms a part of some of these basic chemical reactions. For example, hydrolysis. So hydrolysis, you start out with a polymer of some sort. Here we just have a simple dimer, and you start out with a water molecule. So what happens is enzymes are going to help with these reactions happen as well. They're going to split apart the monomers. They're also going to split apart a water molecule into its component hydrogen and hydroxide ions, and then they're going to join those to the monomers. It's kind of like when we were doing that activity where we were building monomers and polymers, and you can't have any open bonds. You have to have all of the valence shells filled of the electrons, orbitals, and you have to have stable molecules. So in this way, by adding the H and the OH to those open bonds on a monomer, you've done the equivalent of covering up those bits of Velcro. You've done the equivalent of closing up any open bonds. So you may have noticed hydrolysis and dehydration synthesis are basically the opposite reactions of each other. They're very similar to the relationship between photosynthesis and cell respiration. The products of one end up becoming the reactants of the other. And this can keep going on and on. So in this way, your body can take in polymers from the food that you eat, break them down into their constituent monomers, and then take those monomers to rebuild different molecules. That's the way that you can take in protein from the food you eat. You break it down into amino acids, and you can put those amino acids together to make a completely different protein. So now that we've talked about how we build and break these biomolecules, let's talk about each individual type. So carbohydrates, we know they're sugars, starches, things like that. They're a very immediate energy source, but they also have a structural role as well. And when we talk about the various types of polymers of carbohydrates, we'll talk a little bit more about that structural role. Carbohydrates do serve more of a function than just being an energy source. We will run across that theme throughout the school year. They do contain carbon and hydrogen as well as oxygen, and they always contain it in the same ratio. Basically, for every carbon atom, there is going to be the equivalent of a molecule of water. That is literally what carbohydrate means. It is hydro, meaning water, carbon, so it's watered carbon. The different terms we use for anything you see with saccharide in it is going to be associated with a carbohydrate. So a monosaccharide is a monomer, a disaccharide is a dimer, and a polysaccharide is a polymer of a carbohydrate. So monosaccharides are also called simple sugars. The two most common types that you'll run across are hexoses and pentoses. Hexoses are important because a lot of the sugars that we intake for nutrition are going to be these 6-carbon sugars. They tend to have two major forms. They can either be these long chains or ring structures. For whatever reason, we tend to have the ring structure more common in organic molecules. And it also makes for very easy polymerization. So that's why you'll usually see something like this when you're looking at a glucose molecule, often represented as a hexagon. A pentose, on the other hand, is a 5-carbon sugar, and the biggest, most important examples of those are ribose and deoxyribose. Another example is actually, I've got this wrong. My apologies. Fructose is actually a pentose. Nobody caught me on that last school year. Disaccharides can be important as well. So sucrose is general table sugar, the kind that you would get, in a bag of sugar, pour into some iced tea or coffee. Lactose is the sugar that's found in milk. And I just wanted to point out that the bonds that hold monosaccharides together are called glycosidic bonds. They're also sometimes called a glycosidic linkage. But hopefully when you see glyco, you think of sugar. And if you have trouble thinking of that, think of glucose. Glucose and glyco come from the same root word. You're going to see terms later on like glycoprotein, maybe even a glycolipid, and you should know that that means there's sugar involved. So there's going to be a carbohydrate there somewhere. Now, polysaccharides can take a variety of shapes. There are some that are used for energy storage. The two that are pictured here, which are starches and glycogen, are both used for energy storage. Starches are used primarily in plants, and glycogen is used primarily in animals. So when you are eating something like a potato or some bread, you're taking in starch. Your body will break that down into individual glucose molecules, and then if you've got too much of that floating around in your blood, it'll take those glucose molecules and bind them together to form glycogen. There are other important polysaccharides though. So the last 3 polysaccharides on this list, cellulose, chitin, and peptidoglycans, are important in the structures of various organisms. The cell walls of everything except for animals, because animals and some protists are the only things that lack cell walls. All of those cell walls tend to be made of some sort of complex carbohydrate structure. So cellulose makes up the cell walls of plants, and it's a polysaccharide. Chitin makes up the cell walls of fungi, as well as the exoskeleton of some like insects, or also maybe some of the shellfish as well. And chitin is basically, I say, what makes an insect crunchy when you step on it. And peptidoglycans are these complex carbohydrate protein Frankenstein's almost, a mix of different biomolecules that are found in the cell walls of some bacteria. In fact, they're a common target of things like antibiotics or drugs to target bacteria. Now, let's talk lipids. So, lipids are unique amongst biomolecules. While all the other biomolecules have a distinct monomer, and they have these similarities in their structures that let you look at a diagram and kind of identify basically what type of biomolecule it is, lipids only have one thing in common. The thing they have in common is that they are all nonpolar. None of them dissolve in water. Because of that, you can actually have multiple different types of structures that are going to be nonpolar. One of their hallmarks, they have very, very few oxygens, a lot of carbon-hydrogen bonds. They serve as long-term energy storage very often. They can be used in communications in cells, and if you're thinking, wait, how can fat be used to communicate? That's actually the branch of lipids called steroids. They are often functioning as hormones. And they can also serve to protect against dehydration. Waxes, for example, do that. They're an important component of the cell membrane, and they are also involved in cell communication and regulation within the cell. So this is kind of your generic class of lipids called triglycerides, or sometimes called triacylglycerides. If you see the tri and the glyceride, that's you're looking at a lipid. Notice that there are very few oxygens in here. In fact, the major oxygens in the complete triglyceride are right here. However, the majority of these, the bonds that are here and the majority of the atoms that are linked to the carbons are going to be hydrogens, and that's what makes these nonpolar molecules. So a single glycerol molecule gets bound to 3 fatty acids. They're joined by something called an ester bond. So if you see any mention of an ester bond, there's about a 50-50 chance that it refers to a lipid. We'll talk about the other 50% chance that there is. And you may have heard of saturated versus unsaturated fats. The major difference between them is saturated fats have every single bond that can be occupied with a hydrogen occupied. Each carbon is joined to every other carbon by single bonds, single covalent bonds. On the other hand, unsaturated fatty acids are going to have at least one double bond between carbons in their carbon chain. So notice, instead of having a hydrogen coming off here and a hydrogen coming off here, you have not completely saturated that fatty acid with every single hydrogen you can stick on. The big difference that this makes is an unsaturated fat, the fatty acids are bent or kinked. And so they are not going to compact as much, and they're not going to be able to be squished into a small space. This is important, and this is why it's important for health that you eat more unsaturated fat, because saturated fat, you'll notice, it has this very regular, rigid shape. You could imagine stacking a bunch of those together. Since they are nonpolar, they're repelled by water. So if you've got a lot of these saturated fatty acids floating around in your blood, well, your blood is a watery solution, so they're going to be excluded from that solution. They'll clump up together, and since they compact so nicely, they can form these nice, unhappy clots in your blood vessels. Saturated fats tend to be solid at room temperature, and unsaturated fats tend to be liquid at room temperature. That's why you'll get oils, especially from plants, and they're going to be liquid, able to pour at room temperature. Phospholipids are an extremely important modified triglyceride. They do contain a glycerol, and they do also have 2 fatty acids, but instead of having a 3rd fatty acid attached, they have that all-important phosphate group. The importance of this, we'll discuss this in more detail when we talk about membranes and when we talk about cell transport in our next unit, but suffice it to say that phospholipids are kind of bipolar in that half of them is polar, half of them is nonpolar. So the phosphate gives this molecule polarity, but the fatty acid chains are nonpolar like any other fatty acid chain. Because of this, when you stick phospholipids in water, their nonpolar tails tend to clump up together because they are excluded from water, whereas the polar parts of them, those phosphate heads, face outside of this cell membrane or a layer of fatty acids, a layer of phospholipids, and they're going to face the water that's both inside and outside of the cell. And this is a spontaneous formation. You can take a bunch of phospholipids, stick them in a jug of water, shake it up, and they will spontaneously form these structures that will keep all of the nonpolar tails away from water. The other major category of lipids are steroids. So steroids are made of these fused carbon rings. The kind of basic steroid is cholesterol. Cholesterol is very important as a component of your cell membrane. It helps keep your cell membrane fluid so it doesn't stiffen up. Notice that there is only one oxygen in this entire molecule, so that means it's very highly nonpolar. However, steroids, like cholesterol, are the beginnings of important chemical pathways that create a lot of hormones. So a lot of the sex hormones like testosterone and estrogen are steroid molecules. That's why if you talk about an athlete doping, they may be taking steroids, generally testosterone to improve their performance, or a testosterone-like compound. And you can see an example of that here. So here we have a cholesterol molecule, and if you modify it in a certain way, it becomes testosterone. Put that into an adult mammal, and it will start to develop male secondary sex characteristics, like this giant floofy peacock here. On the other hand, if you modify it in a slightly different way, you'll create estrogen, put that in an adult animal, and you'll induce secondary female sexual characteristics, like the kind of less startlingly beautiful peahen. Don't worry, she's got some pretty feathers too. Waxes are another type of fat. They're made of a fatty acid, but they've also got an alcohol. They're also solid at room temperature. Their importance is waterproofing. So if you've ever picked leaves off of a tree or a plant and kind of scraped your fingernail across the surface, you may have gotten some waxy buildup under your fingernails. That is a protective layer of wax that plants have to prevent themselves from dehydrating. Beeswax is going to help protect the honey and the larvae in the hive, and earwax helps protect those delicate structures in your ears. It's also why you wax a car. Proteins are by far the most varied and most diverse group of biomolecules. The thing they have in common is that they are all made of amino acids, and these amino acids are linked to each other by peptide bonds. Anytime you see PEPT, P-E-P-T, that means you're looking at something related to proteins. So, for example, peptidoglycans, that peptid part, has to do with the fact that there are some amino acids in that molecule. A peptide is made when you bond a couple of amino acids together. A polypeptide is when you've got a long chain of amino acids bound together. And once you take that polypeptide, fold it into a particular shape so that it's able to do a certain job, it becomes a protein. Proteins function in almost everything. The only major functions in the human body, or any body, that they don't perform are energy storage and carrying genetic information. Proteins are so integral to the function of the body that your body will not, let's say you're starving, you're on a deserted island, your body will break down everything it possibly can before it will start breaking down proteins for energy. If you start breaking down proteins for energy, you are very near death. So, a very major category of proteins are enzymes. We're going to talk about them a lot more in our next unit. They also form structures within the body or within cells that help keep shape. And similarly, they're also involved in motion. They can move things around either into or out of a cell like a membrane protein, or around the body like hemoglobin. Antibodies are a particular type of protein that are used in our immune system. And then you can also have protein-based hormones that can act as messengers between cells. So this is the basic structure of an amino acid. Within living things, there are about 20 amino acids that are found. You do not have to memorize the names of them. You do not have to memorize the structures of them. What you need to know are basically the things that you see here. Amino acids are made of a central carbon atom. It's got a hydrogen coming off one side. It's got an amine group coming off another side. On a third side, it's got a carboxyl group. And then the last bond that central carbon is forming is going to vary between different amino acids. We call it the R group. That R is like a variable, and it's standing in place for anything that you can join there. Some R groups are super simple. The simplest amino acid just has a hydrogen. That's glycine. On the other hand, some of these amino acids have very large R groups that have like ring structures and all sorts of different atoms in them, like sulfur, and they're obviously going to have different properties. But the most important thing to remember is this general structure. If you see nitrogen and you don't see a ton of rings, you're looking at a protein. So here are the 20 major amino acids. Like I said, you don't have to memorize them, you don't have to know their structure, but what I do want to point out is that we can divide them into these groups. So this chart is basically showing that you can have some that are aliphatic, aromatic, acidic, basic, hydroxylic, containing sulfur, amidic, and then you have your essential and your non-essential amino acids. All of those terms that I just said basically are referring to the reactivity or the interactions that these amino acids have with other molecules. This can confer polarity. This can make these amino acids prone to forming acids or bases, and it determines how they'll interact with molecules around them. Depending on which R group you have, you'll have a polar or a non-polar amino acid, and this is going to become really important when we talk about protein folding. That's just showing you a dehydration synthesis reaction and the formation of a peptide bond. Let's talk about protein structure. Proteins have to have a very particular shape in order to fit their function. Just like any tool has to have a certain shape in order to do its function. A hammer wouldn't work if it was shaped like a pair of pliers, not for the same task. When proteins do not have their proper shape, when something has happened to disrupt their shape, we say they become denatured and they are no longer functional. If it's a serious enough denaturing, this could be enough to completely destroy the protein and it will not work again. The things you can do to denature a protein are change the pH, change the temperature. Those are the two big things. And the more damaging temperature aspect is going to be raising the temperature. This is why it's dangerous to have a very high fever. You're basically cooking your proteins from the inside out if your fever gets too high. So we're going to look at the 4 different levels of protein structure next, because they help determine the 3D shape of a protein, which helps determine its function. So you start out at the most basic with the primary structure of proteins. That is just the chain of amino acids, their identity of them, and how they're arranged. So they are usually, you'll have like G, V, S, L, T. Just think of each of those letters as representing a particular amino acid. And that sequence of them is the primary structure of a little protein that we're thinking of. Based on the functional groups that are on the R groups on these amino acids, they're going to form these secondary structures that are called either alpha helices or beta pleated sheets. And these things are held together with hydrogen bonds. So hydrogen bonds have a huge impact on the 3D shape of molecules. Then the tertiary structure is going to be those alpha helices and those beta pleated sheets folded on top of each other into a final shape. They're stabilized by what are called hydrophobic interactions, which we'll talk about in a moment. By hydrogen bonding, there can be some ionic bonding, so attraction between ions, that's called a salt bridge. And then there can also be covalent bonding like a disulfide bridge. And this is a fairly stable structure. There are some proteins that have a quaternary structure, and what that means is they've got more than one polypeptide chain, so more than one chain of amino acids that are associated with each other. They can be covalently bound to each other or held together by these other non-covalent bonds. So here we see an example of all 4 levels of structure of an example protein, which happens to be hemoglobin. So hemoglobin's primary structure you see is almost like a chain of beads. Those are the individual amino acids, the sequence of amino acids that make up this protein. And that sequence is determined by the instructions in your DNA. So notice you have peptide bonds between the individual amino acids. Also notice that you have these pink regions that are highlighted. Those pink regions, based on the R groups and the sequence of amino acids, form either an alpha helix, which just looks like a curl, or they form a beta pleated sheet. Beta pleated sheets are often represented like this one right here. in 3D structures of proteins. You'll see what's called a ribbon model very often, which shows a protein like the primary sequence is this long ribbon and it looks kind of folded up to form this complex three-dimensional shape. And then hemoglobin happens to have 4 different subunits, so 4 separate peptides that join together to form a mature hemoglobin molecule that's capable of carrying oxygen around in your blood. Now, if it's a simpler protein that doesn't have multiple peptides and it's just a single chain of amino acids, it won't have a quaternary structure. It'll stop at tertiary structure. And if I have thoroughly confused you, well, that's why you have that protein structure packet. And that's also why we're going to do an activity with proteins in class. So there are some interactions between the R groups that help stabilize these 3D shapes. I talked about disulfide bridges. Those are when 2 sulfurs on 2 different amino acids will covalently bind to each other. That forms a very strong bond, and that is a very heavily stabilizing influence. Hydrophobic side chains tend to kind of bunch up together on the interior of proteins, and the reason for that is they are excluded by water molecules. When I'm personifying these things, I tend to say that water molecules are chargist. They don't like to interact with things that are non-polar. They'll only interact with polar things. So water molecules will kind of be attracted to each other, and they will exclude anything that's non-polar. So if you have a protein with a bunch of non-polar pieces, say right here and right here, that protein is going to fold up so that those regions are as far away from water as they can get. Then you can have these ions that are charged that are basically electrically attracted to each other, and those form what are called salt bridges. Proteins can be modified, so you can actually do some covalent bonding of other molecules or of other groups to the R groups on an amino acid, and that can be used to change the shape. When a protein forms a temporary association with another molecule, that can also change its shape, and that's very important when we talk about enzymes. So for example, you have this molecule right here. When it bonds to this protein, not a covalent bond, not a permanent bond, forms a hydrogen bond. That can change the shape of the protein. In the case of enzymes, what would happen is the changed protein would then modify that molecule that's bound to it. And then once the bound molecule releases, it goes back to its original shape. You can also add modifications onto proteins to temporarily inactivate them or activate them. And that gives your body and cells a level of control over when proteins are active or not. But just like it's essential for proteins to be folded correctly, there are also diseases where mistakes happen and proteins are not folded properly. There are chaperone proteins, proteins whose entire jobs are to help proteins fold correctly, and when there's a problem with them, that's when you can develop diseases. So there are hints that there are links between defects in these chaperone proteins and diseases like Alzheimer's disease and cystic fibrosis. Then hopefully none of y'all will ever encounter prions. Prions are actually these misfolded proteins that are infectious. So what happens is if you ingest brain tissue of the same species, so let's say you're a human cannibal and you're eating some brains of another human, what can happen is these prions can end up getting into your body, and then they can change the proteins in your own brain and basically form these plaques and cause these diseases called transmissible spongiform encephalopathies. And the most common example of that is mad cow disease, because once upon a time people decided to feed ground up bits of cow to other cows. Now, let's talk about nucleic acids. We're not going to spend a lot of time on nucleic acids right now because they're going to get their entire own separate unit later on. So let's just talk about their basic structure. A nucleic acid is made up of nucleotides. Those are the monomers of a nucleic acid. And the bonds between these nucleotides are called phosphodiester bonds. That's what I was talking about when I said if you see an ester bond that you're looking at a lipid. That's not always the case. If you're looking at a phosphodiester bond, then you are looking at a nucleic acid. The important way that you can distinguish nucleic acids, they're going to have a ribose or a deoxyribose, so they have a 5-carbon sugar. They've got phosphates, and then they've got this nitrogenous base sticking off the side. And the shapes of these nitrogenous bases will depend on what diagram you're looking at. Sometimes they're these simplified shapes that you see here that kind of fit together easily. Other times you'll see representations of their actual chemical formulas. Their most important task is, of course, carrying genetic information and also contributing to the formation of proteins. We're not going to talk about DNA and RNA so much right here. Instead, we'll talk about ATP a little more. Notice that you have hydrogen bonds between the nitrogen bases. So the only thing holding together two strands of DNA are a lot of these really weak hydrogen bonds between molecules. So each individual hydrogen bond can be really weak, but you get like thousands of them holding two molecules together, and they can hold them together pretty strongly. So bases pair with each other. Adenine always base pairs with thymine because they can form the correct number of hydrogen bonds with each other. And cytosine always base pairs with guanine because they can form the correct amount of hydrogen bonds with each other. And of course, in RNA, thymine is replaced with uracil. So, while you have a very characteristic shape to DNA, the double helix, RNA can actually form a lot of different shapes. If you'll remember, RNA is single-stranded, usually. However, there can be areas of RNA that are complementary or matching to other segments of RNA, which can cause RNA to fold, and then it can hydrogen bond to itself to form these kind of complex 3D structures. And we'll talk about that more when we get into genetics. A couple of important things about nucleotides. They have directionality. Just like you read language, we read English from left to right. You actually read DNA and RNA in a specific direction. So DNA and RNA are always read from what's called the 3' direction to the 5' direction. The 3' direction has a hydroxyl group hanging off the end, like what you see here, and the 5' group has a phosphate hanging off the end. So if you want to think about it, to help you remember, the 5 phosphate. So the F sounds go together. So 5' has a phosphate, and the other one's the hydroxyl, and the other one's the 3'. However, DNA and RNA are not made in the same direction that they're synthesized. Instead, they're made in the 5' to 3' direction. And this may not seem very important right now, but it will get more important when we start looking at DNA replication later. So nucleic acid strands are complementary to each other, meaning they match up with each other when they base pair. They are also anti-parallel. So if you've got two DNA strands, they are read in the opposite direction. So this one would be synthesized in this direction, the 5' to 3' direction, and the opposite strand, the complementary strand, would be synthesized in the opposite direction. So you wouldn't actually read the two strands of DNA going in the same direction. And then finally, ATP. I know you guys know it's the energy currency of the cell, but try to avoid putting that on the AP test. It stores chemical energy, or it is a readily available source of chemical energy, the immediate source of chemical energy for reactions in the cell. It is a modified phosphate. It's got an adenine, it's got a ribose, and it's got 3 phosphates on it. Those phosphates are super important. Basically, phosphates are very highly polar, they're heavily charged, and it takes a lot of energy to get 2 of those same charges. They're both negatively charged, so negatives and negatives repel. It takes a lot of energy to force them in proximity to each other. So if you've got 2 phosphate groups bound to each other, they're not super stable, and there's a lot of energy involved in making a phosphate-phosphate bond. Similarly, or conversely, if you break a phosphate-phosphate bond, like what you see happening here, that releases a lot of energy. And so your cells have found ways to use the energy released by breaking these phosphate bonds to power reactions in the body. So that is it for our organic chemistry lesson. We'll have some other ones later.